Morning, everyone. My name is Samantha Riles, Communications Officer from Sugar Research Australia, and I'd like to welcome you to today's webinar. This webinar is the third in a three-part series focusing on biosecurity. Our presenters for this series of webinars are Dr. Rob McGarry, Dr. Nicole Thompson, and Dr. Kevin Powell. In today's webinar, Dr. Kevin Powell will be presenting about pests, major threats, and current research. Please note that today's meeting is being recorded. Please keep your microphone on mute to keep the background noise to a minimum. I will be assisting as a co-host to ensure that all runs smoothly. If you are having any technology issues, please send a message in the chat box, which is located on the menu bar of your screen. I have included details in the chat box of how you can dial in via phone if you're experiencing computer issues. We encourage you to take an active part in today's meeting by asking questions. You can do this at any time throughout the meeting by typing your question into the chat on the menu bar. I will ask these questions at the end of the presentation on your behalf. If it is easier for you to ask your question verbally than to type in the chat, please unmute yourself. And I'd now like to hand over to our speaker for today, Kevin Powell.
Good morning, everyone. I'm Kevin and I'm based at SRA Moringa. I'm relatively new to the sugarcane industry. I joined SRA in 2017, but I'm not new to pest management. And a particular focus of mine has been on root-feeding insects and sap-sucking insects. I'm also not new to working with endemic and exotic biosecurity pests, and I've worked in several countries on a range of pests, which you can see in those pictures there. Before we get into the talk, I just wanted to highlight some of the terms I'll be using. So the first term is endemic. And what I'm talking about there is native insect pests. And a good example of that in sugarcane would be soldier fly. So an exotic pest is a pest that we don't have, but it could be accidentally introduced. And a genotype is referring to the genetic characteristics of a particular insect as denoted by its DNA. The outline of my talk, I'll be first talking about endemics, i.e. which pests we have, exotics, what we don't have, recent incursions, what's recently entered, and the biosecurity approaches for pests coming in. And finally, I'll just be talking about innovative approaches to looking at these pests. So with endemic pests, the sugar industry is quite lucky in a way and unlucky in other ways. So we have two really main pests of sugarcane, and they're both root feeders, so soldier fly and cane grubs. They do cause extensive damage, but we don't have the number of pests that other industries have. We also have some sap-sucking insects like Perkinsiella, which is a vector of Fiji leaf gall. A number of projects we've had on those pests have recently been completed. So why am I talking about exotics? Well, really exotics have been a problem for all agricultural industries, and I've listed down there on the left-hand side of the screen a number of exotics which have come in. And they've come in mostly in the last 25 years. You'll see the first pest in the list there is phylloxera. That's a pest which I will refer to occasionally through my talk. That's actually been here since 1877, and it's one of the few pests which is effectively controlled by quarantine. Most of the other pests that have entered haven't been able to be effectively controlled by quarantine, except for potato psyllid. And they've become established, and eradication has proved not effective. The only effective eradication technique that I've seen from the pests on that list is the eradication of phylloxera in Queensland. And that was basically eradicated by ripping up the vineyards which phylloxera was attacking, and those vineyards are now covered with the Brisbane airport. So concrete is a really good way of getting rid of some pests. At the bottom there, you'll see the fall armyworm, which came in to Australia. It was detected in 2020 in Queensland. It's also been detected in the Northern Territories and WA, so it's now established. When we're talking about major exotic threats to the sugar industry, there are actually too many to list. So I'll only be referring to two groups, the Lepidoptera, or the moths, and the Hemiptera, so the sapsuckers, particularly focusing with the sapsuckers on leafhoppers and planthoppers, because they're important vectors of things like phytoplasmas and viruses. We have projects in all of those three pest groups, except for the Hemiptera, and that's an ACR project which was recently completed, and I'll give some examples of that work. So if I focus first on the sap-sucking insects, what are the threats? Well, the threats really lie with a planthopper, which can transmit the virus which causes Ranu stunt disease. You can get extensive losses with Ranu stunt, which has been seen in a neighbouring country, Papua New Guinea, where they've had up to 60% loss of the crop because they were using a highly susceptible variety. In Australia, we do have a good level of resistance in our varieties. We don't have the Ranu stunt virus yet, but we do have the planthopper present up in northern Queensland. So it's really important that surveillance takes place, and this is happening up in the north. And also we have a very good diagnostic technique for the virus. Another important threat is white leaf disease. This is caused by a phytoplasma, and the vector here is not planthoppers, it's leafhoppers. So these two vectors are found overseas in Asia. Also, one of the vectors is found in Papua New Guinea. The phytoplasma is transmitted by at least these two leafhoppers. It's also been detected in 12 other species. It's mainly controlled using resistant varieties, and in some countries, hot water treatment, although the efficacy of that is a bit uncertain. Another virus which we have been doing some work on in Indonesia and Myanmar is the sugarcane streak mosaic virus. Now, we don't know which vector is involved in transmission of this virus, but I recently completed some work in Myanmar, which I will talk about. It's mainly controlled with resistant varieties. So I'm now going to go through some of the biosecurity approaches that all have to be followed if you're going to get effective management of an exotic. So I'll go through each of these particular subject areas and finish up on innovation. So first, detection. When you're looking at detection, it can sometimes be like looking for a needle in a haystack. It can be very difficult, particularly if you're looking for detecting below ground pests. Detecting above ground pests is somewhat easier because they're quite visible, but with below ground pests, you're sometimes really looking for the damage which is seen in the canopy. So I'll give you a few examples of how we detect above ground pests. And this is an example of the Mexican rice spora, which affects rice and sugarcane in the US. Now, this particular borer comes from Mexico, obviously, and it crossed the border and was detected in Texas in 1980. And pheromone traps were used, and it was then detected in Louisiana in 2008. So it was gradually spreading through the country. They use male pheromone lure traps to detect it. This is something that's also used for armyworm, and they use this as now as a crop monitoring surveillance technique. To manage Mexican rice borer, they also have quarantine, they have some resistant varieties, and they use insecticides, but only sparingly when the populations of larvae reach their economic threshold. So a bit more on above ground pests. Since I started at SRA, I've been experimenting with a number of standard entomology techniques for trapping potential vectors of pathogens. So vacuum traps, sticky traps, water traps, and a standard sweep net. Now, with these trapping methods, they're not as specific as male pheromone lures that we use for moths. So they do require more sorting, but they're highly effective in catching a range of target groups. And one of my target groups in the north was Mealybugs. And you'll all be familiar with the pink sugarcane mealybug, which is generally not a major issue, but it's present in most sugarcane crops. And it's common to most regions where we grow sugarcane. What I actually found in the north when I was sampling was another mealybug, this white mealybug on the right-hand side there, which instead of attacking the stem, attacks the leaves of the sugarcane plant. Now, we haven't done any studies to look at the extent of potential damage on these, from these insects, but we do know that this insect has not been reported on sugarcane in Australia before I detected it, but it is known on sugarcane in other countries. And the insect is present and has been present in Australia since 1926, where it attacks paspalum, and it might be associated with pastute dieback in paspalum. Some of these techniques that I've been using are transferred across to a study we did in Myanmar. This was essentially a 5-month study where we were trying to find what the potential vector of sugarcane streak mosaic virus was. So I went across to Myanmar in February last year, and I was assisted by a whole group of students and researchers and set up my trapping methods, trained them in how to use those trapping methods to see what potential vectors we could find. We had 2 trial blocks. One was with a Chinese variety, and one was using an Australian variety Q208. These blocks were relatively close together, and what we saw was that sugarcane streak mosaic virus symptoms were high in the Australian variety and not visible at all in the Chinese variety. So I monitored various insect groups using the different trapping techniques. And this is just showing what we caught in the yellow sticky traps over a 5-month period. And what I just wanted you to look at here is the leafhopper numbers. So you can see by the red arrow there that the leafhopper numbers were extremely high on Q208. So I decided to take a look at the identity of those leafhoppers and also do some virus testing on the leafhoppers. We found more than 30 species of leafhopper, most of which are not found in Australia. And we also found 4 new, previously undescribed species. 8 of the species that we found turned out to be sugarcane streak mosaic virus positive. 3 of these species actually do occur in Australia, and they are known to be associated with other viruses. At this stage, we don't know which species actually transmit the virus, and this work needs to be continued to determine that. Detection of below ground pests can be done in a number of ways, and I've just put there 2 industries, sugar industry and the viticulture industry, and shown which detection methods have actually been used. And you can see in viticulture a whole range of detection methods have been used for a below ground pest. In the sugar industry, there's not quite so many techniques have been used. At the end of the talk, I'll hopefully be able to have time to show you a sniffer dog trial, which I did in my previous job, working on the grapevine phylloxera. And in a minute, I'll talk about cane grubs and the work that's been done using multispectral sensors. There's 2 types of sensor you can use for aerial sensing, multispectral and hyperspectral. The hyperspectral gives you much more detail than multispectral because it has more spectral bands to examine. And you can use those sensors on satellites, on aeroplanes, or on drones. So satellite imagery has been conducted as part of a cane grub experiment where we were seeing whether we could identify risk areas for cane grubs. If you can identify risk areas early, you may be able to manage those areas differently. So 2 satellite systems were examining, the GOI and the Spot 6. Each of them has their advantages and disadvantages. Overall, though, we found that this is not really a good system for early detection because you get a lot of false positives. So we're not exploring this at the moment any further. Another approach, which again has been taken in the viticulture industry, is to use drones, and this was some work which I did a couple of years ago in the Yarra Valley. And we had drones using different types of camera, including hyperspectral. And what we found on the matchstick there, by the way, is the size of the insects. So they're quite difficult to detect. What we found was that using a UAV with a hyperspectral camera, you could pick out more spots where the insect was present, as highlighted by the red patches in that bottom diagram. So having detected an insect, you need to be able to identify what it is. And this is mainly done through morphological techniques and DNA barcoding. So we have had a project working on exotic stem borers, and I'm showing there the top 7 high-risk exotic threats of stem borers coming into the country. And they're located in Asia and Papua New Guinea. We have taken samples from various collections and conducted a study on those samples, and that just shows you where some of those collections were taken from. And we used DNA barcoding in the first instance. Why would we do that? That's because identifying these moth borers is very difficult, both in the adult and the larval stage. The only way you can really identify the adults properly is to do dissections of their genitalia, which is quite laborious, and you have to be an expert to do that. We've used 2 approaches, DNA barcoding and genome skimming. I don't have time to talk about the genome skimming, but essentially we take specimens and remove part of the insect and look at its DNA. And also we then look at the genetic characteristics of that and compare that with known barcode sequences in gene banks. With this work, we've looked at 64 different species, taken 500 plus specimens, and looked at over 1,200 sequences. And when this work is completed, we will have good diagnostic, morphological and molecular protocols for the pests. And we will improve the current SRA pest dossiers and distribution maps. And when necessary, we will split species from 1 species into 2 species. Once you've actually detected a pest, how do you control it? Well, there's various ways, including resistant varieties, IPM and chemical control. With the moth borers, we're getting ready in case there's an incursion. So we're doing trials in Indonesia and Papua New Guinea using insecticides. The trials are working extremely well so far, but the project is currently obviously delayed because of COVID-19 restrictions. Just now touch on fall armyworm, which is now established. There are lots of knowledge gaps for fall armyworm in Australia. The biggest knowledge gap is we don't know if there's going to be any major damage to sugarcane here. In Asia, the damage has not been extensive, and it all really depends on which strain of fall armyworm you get. And the strain of fall armyworm is also really important because you could have insecticide-resistant strains. We need to know, if you use insecticides, how is that going to impact on natural enemies of fall armyworm and the other armyworms that we currently have? And we need to have good diagnostics. So essentially more work needs to be done on fall armyworm to determine what it will do to sugarcane here. And we already have endemic armyworms. So they're not generally managed. And if you use insecticide sprays, it really disrupts the ecology of the natural enemies. So we really don't recommend insecticide spraying unless it's absolutely necessary. So what has SRA done so far in the fall armyworm space? Well, we applied for emergency use permits as soon as we knew that fall armyworm was in the country. So there are chemicals available for control, but the really important thing is you need to first identify if what you've got is fall armyworm or if it's one of the common armyworms. And we're currently also linked to a project with CSIRO and GRDC examining the strains of fall armyworm that are here, and also examining their insecticide resistance status. When a pest comes in, you always try and restrict its distribution. This is not always possible. It depends how quickly the insect adapts and how quickly you detected it. But you can reduce the spread by the use of quarantine zones. And you'll all be familiar with the quarantine zones that we have for the sugarcane industry, and those zones work very effectively for things like Fiji leaf gall virus. Quarantine zones have been used in Australia for quite some time. In the viticulture industry, they've actually been used since 1890. And this just shows you a quarantine map for phylloxera, which was first detected in Victoria and has not spread much further than Victoria, apart from a couple of little outbreaks in New South Wales. So highly effective for certain pests. With that particular pest, they also have protocols to reduce the risk of spread out of the quarantine zone. And those protocols cover machinery, planting material, etc. To restrict the spread of an insect, it's also important in some cases to use disinfestation techniques, and you're all familiar with the hot water treatment for sugarcane. In the grapevine industry, they also use a whole range of different techniques for disinfesting harvesters, machinery, footwear, etc. And they even use disposable coveralls when working in an infested zone. Just finishing off now with innovation. We know that there are lots of standard techniques for detecting and managing biosecurity pests. But we also need to keep on top of our game with other approaches. So these are some of the novel approaches. So I'm now going to just jump to a bit of genetics. It's really important when an insect is found that you know the genetics of the insect. I'm showing you here some recent genetic studies done on soldier fly larvae. Now, we know that there are two species of soldier fly larvae. So we did some genetic work on soldier fly larvae from different regions. And when we actually looked at that, we found that there were basically 5 different genetic clusters of soldier fly. Now, you wouldn't really expect so many clusters if you only had 2 species of soldier fly. So we looked at the adults of soldier fly, And you'll see there are two species, Anopus rubiceps and Inopus flavus, which we all know are the sugarcane soldier fly and the yellow soldier fly. We actually looked at 36 adults from different regions. We sent them to an expert taxonomist who looked at these soldier flies, and he identified them and said, you have 4 new species of soldier fly. So we don't have 2 species, we actually have 6 species. So why do we care if we've got 6 species of soldier fly? Don't you manage them in exactly the same way? Well, I'll give you an example here again, which shows some of the work I did in the viticulture industry. What we looked at there was genetic clusters of this pest phylloxera, root-feeding pest, like soldier fly is a root-feeding pest. We looked at 6 genetic groups, G1 to G30, and tested them on how badly they damaged different varieties of grapevine. And what we found was only 2 of those groups caused high damage, and 4 of them caused low damage. And 2 of those groups, you could use resistant varieties, but for most of those groups, you had to use tolerant varieties. So the pest was still there, so you still had to use quarantine to manage it. Another technique we've been using is called the EPG, and this has now been adapted for sugarcane pests. The EPG is basically a simple electronic system where you wire an insect and you wire a plant and you send a small electrical signal through the insect. This is not meant to electrocute the insect. This is actually meant to monitor the feeding behavior of the insect as its mouthpart moves through the plant. So why is this useful? This is actually extremely useful technique because you can see how well insects feed on the plant. So if you've got a plant which is highly susceptible, the insect will spend a lot of time feeding in the phloem. If you've got an insect which is a plant which is resistant, the insect does not spend very long in the phloem at all. So you can use this for varietal screening. You can also spray insecticides and see how that affects feeding. And you can look at transmission and acquisition of pathogens like viruses. Initially, we're working on Perkensiella, but I'm hoping that we will be able to use this technique on exotic vectors overseas, and it can be used for any sap-sucking insect. We've also been involved in the IMAPS Pests of Sentinel surveillance project, which Nicole mentioned in her previous talk. And this is all about improving diagnostics and improving sampling for pests. So we've had the Sentinel Pest Detector up in Moringa for about a month, sampling insects. Using that very tall tower there, you can sample insects at different heights. Those samples were all collected in March. They were sent off to SARDI in South Australia and AgriBio in Victoria. They're doing some DNA work on those samples, and once they've completed that, they'll send reference samples back to us so that we know what was caught in those particular traps. The other trap sampler we're looking at using later on this year is the fall armyworm automatic sampler. This sampler actually detects fall armyworm as they come into the pheromone lure trap. So pretty excited about being able to test the efficacy of that trap system. So there's been a lot to take in that presentation, so I hope I haven't lost you all. But in summary, I just want to say that we are very fortunate in that we have few major endemic pests, but unfortunately there are multiple threats out there that we need to be prepared for. Unfortunately, also a lot of the research has to be conducted overseas, which is challenging when you have COVID-19 restricting travel, but you do need to work overseas to work on exotics. Biosecurity has many different aspects. Quarantine is absolutely essential, as is a good detection system. And you have to be able to diagnose your pest accurately, and management will be different between different pests. And innovation has to happen in order to keep on top of our game. So we can prepare for high-risk pests, but you have to be prepared for the unexpected. And something can come in which you are not quite prepared for. So we do have to be prepared for the unexpected. So thank you very much, and thank you for listening.
Thanks, Kevin. So you'll notice that I put a link to a survey in the chat box. Your feedback is important to us, and I encourage you to complete that so that we can improve on our presentations and webinars. Please type any questions that you have for Kevin about today's webinar topic into the chat box, or unmute your microphone. Please also include any further questions for Rob or Nicole, our presenters from our previous biosecurity topics, as they're both online. And as Kevin mentioned earlier, depending how we go with question time, we've got a few short videos that we would like to show as well. So I'll just jump into the chat box and see. Got some questions here, Kevin. Harjeet's asking, can pheromone traps be used for trapping below ground pests like cane grubs and soldier flies during the above ground phase of their life? Would that help reduce numbers?
Yeah, pheromone traps can be used to detect the above ground part of the life cycle, so the adults. But the very first thing you have to do is identify the sex pheromone from your insect pest. So that can be quite a challenging thing to do. It is problematic with things like soldier fly to actually determine when you're going to get a bad year from soldier fly, and to determine when the adults are going to fly, because they only fly for about 5 days. But I'd say there's potential for using pheromone traps, but a lot of work has to be done in order to first identify a pheromone and then test whether it's going to work for those pests.
Okay, thanks, Kevin. Just waiting for some more questions to jump into the chat box. Please feel free to unmute your microphone if you'd prefer to ask your question verbally. So in the meantime, Kevin, I might share my screen and we might look at the first video.
Okay, yep.
Just let me know if you can see that.
Okay, I'll just talk to this video. What this video is doing, in 2 of these 10 containers, there is a very small insect, 0.3 of a millimetre in size. And this dog has been trained to detect volatiles given off by the insect, and it's going to now move to the second rack of samples. We have essentially the very small insect in a petri dish in one of those containers, and the dog will stop and wag its tail when it reaches the appropriate one. So it's picked it up. I was very skeptical about using detector dogs for such a small insect, but this dog was extremely good at doing it. However, it did have another dog from the same litter who just didn't have the right temperament and couldn't detect the insect at all. So if you're going to use detector dogs, it takes a lot of training, and like humans, you have to get the right person to do the job. Next video. So these two videos are just showing you, in principle, how an insect is wired up for the EPG technique, so you can monitor its feeding behaviour. This is wiring a very large insect, so you've got a shield bug there. Most of the insects I work with are a lot smaller than that, so it's much more challenging to fix a gold wire on the back of the insect with silver paint. But you can see the insect is maybe not happy, but it's dangling on that piece of wire there ready to be placed on the plant. Next video, please. So once you have your insect wired up, what I'm showing you there is the screen where the data will come through when the insect is feeding. You can monitor 8 insects at a time, and this is the amplifier, which actually amplifies the signal from the electronic voltage going through the insect. And these here are that's the probe holding the insect, placing the insect on a plant. And there's also a probe placed in the pot where the plant is. Once you've set all those up, you run the experiment for up to 16 hours, and then you analyse the feeding behaviour over time. And in a minute, you should be able to see a signal coming through when the insect is feeding. So this is a Faraday cage, which cuts out electrical interference, because it's a very small voltage you're working with. And I'm not sure if you'll be able to see that, but that's our screen. And these are waveforms which start to occur. So that insect is now probing and starting to feed on the plant. And each particular waveform, you can characterize and determine if that waveform is in the xylem, phloem, the parenchymal tissue, and work out the durations of those particular waveforms. So it's an extremely sensitive technique.
Okay, I'll just jump back into the chat box. Okay, we have a question from Megan McKenzie.
Yep.
If you had extra funding this year to fund a project that would have maximum impact, what would you do?
That's a very, it's a very hypothetical question, because I'm not sure how much funding we're actually going to get.
Kevin, it's Megan McKenzie here.
Yep.
So I'm from the Department of Agriculture and Fisheries, and so we do some funding through
Okay.
SRA, and I'm quite keen to know if we could put some investment in this area, what would it be? So, and we're certainly looking for impact. So it's just to get a sense of what that could be.
So in terms of endemics or exotics?
Either. It's about impact, really.
Yeah. I think with exotics, we really have to look at our high priority pests and it would be investing in what is going to be the highest risk to the sugarcane industry. So there's a couple of things there. Moth borers, there's been a lot of investment in diagnostics, so we're really on top of that. We have been working on chemical control, but is chemical control going to be the answer if you actually get an incursion and it's been here for a while and becomes widespread? Chemical control is good if you can pick up the insect early and try and delimit it, but as we know with fall armyworm, insects can spread quickly. So I think working on a high priority pest like moth borers would be good for exotics. In terms of endemics, there are cane grubs is our biggest issue, and imidacloprid has been highly successful in controlling cane grubs. But what would happen if imidacloprid was no longer available or its use was restricted? We really don't have any alternative for controlling cane grubs. So I think we need to look very closely about what to do with cane grubs.
Excellent. Thanks, Megan and Kevin. I've got a question here from Priya. Are you developing pheromone traps for potential exotic borers in sugarcane?
No. Is the quick answer for that. I would love to look at pheromone work, but we're not doing any of that work. And that's where I say with exotic borers, it's important not just to focus on things like chemical insecticides. We need to look at other approaches because, as we've seen from my earlier slide with the Mexican stem borer, pheromone traps have proved very effective there for monitoring that particular pest, but we haven't been working on pheromone traps for exotic borers at all.
Okay, thanks, Kevin. Xiaomeng had his hand up earlier throughout the webinar. If you did want to ask any questions now, feel free to turn your microphone on or put them into the chat box.
Gone for a cup of coffee, I think.
Okay, Rob Miller is saying that he supports Kevin's thoughts on cane grub further research. Do you have any comments on, I'm not going to say this correctly, Clothiandian for cane grubs? Rob, you might want to turn your microphone on and correct me if I've got that.
Yeah, no, that's okay. It's the insecticide Shield. That's the active ingredient in Shield.
Yeah.
So it's been a few questions, and it's actually registered for cane grub control, but I have not been able to get my hands on any data to discuss its efficacy. Yeah,
we recently did look at some alternatives to imidacloprid. Some of the data in there is commercial in confidence. But we did, because Shield is registered for cane grubs, we actually looked at that. One of the issues we have in doing insecticide trials is doing trials where you've got high levels of cane grubs present. And the problem with most of the trials was that the controls didn't have high levels present. So Shield appeared to have similar efficacy to imidacloprid, but statistically you couldn't actually prove that because the controls weren't particularly high. We do have a new project, which is an innovation project, which I didn't mention, where we're actually trying to develop a better system for initial screening of insecticides used in artificial diet. So once we have that artificial diet tested, we'll be able to test some of these active ingredients much more rapidly before going out into the field situation. So the short answer is I can't really say if Shield is equally effective to imidacloprid, but it's one of the compounds that we're certainly interested in retesting.
Okay, thanks, Kevin. Another question from Priya. Do you think RNAi or CRISPR, CRISPR, would work against cane grubs?
I really don't know the answer to that one. So I think what you've got to do with cane grubs is look at things which could be cheap enough to do and don't require a lot of investment. So I'm not sure whether RNAi, how much investment that would require to explore the possibilities of whether it would work against cane grubs. But it is a technology that is out there, which may be worth exploring. But we've just got to prioritise what approach we have for some of these major pests. Insecticides, when you get a good insecticide that's highly effective, like imidacloprid, do work extremely well. But it just depends what alternatives there are out there.
Thanks, Kevin. So we will wrap it up shortly. So if you have any questions, this is your chance. A reminder that I put the survey link in the chat box and we'd love to get your feedback. The recording from today's webinar will be available on the SRA website and will be shared via our next e-newsletter. Thank you to Kevin for presenting today. And thanks also to Nicole and Rob for presenting earlier in this webinar series. And we might, we've just got one more question's popped in. This is a question for Nicole from Priya. Regarding the recent threat of foreign seeds being posted into Australia, are you planning to flag this in the regions? Just off mute now, Nicole.
My Teams is a little unresponsive this morning. Priya, are they seeds from sugarcane, or are they seeds from other crops that you're talking about?
Hi, Nicole. Yeah, no, there wasn't mention of exactly what the seeds were, but it was put on ABC this morning that people have been receiving seeds from Taiwan and China and just to hand it into DPI. And I just wondered whether that was going to be flagged, in our sugarcane growing regions as well. It's a biosecurity thing.
Yeah, well, I know that the department, both the federal and the state department, are doing a lot of communications around that. It's probably a good idea that we do mirror that in the regions. I don't know exactly. I know that, for example, the Biosecurity Queensland are
at the moment focusing on Facebook, Etsy and eBay sales of various items. And they're doing quite a lot of communication around that. I don't know of anything specific to seeds. I might have a look at that article, Priya. You just let me know about it and I can follow up with the department to see if there's anything that we can assist with mirroring that communication to the regions. We hadn't planned to do that. Basically what, when it comes to seeds or people buying things off eBay or Facebook or anything like that, especially like sugarcane is something that gets purchased, I know, through like Facebook Marketplace and all sorts of stuff for people to grow in their gardens. And then there are, noxious weeds that are being sold as pond covers and all sorts of stuff. A lot of that, our responsibility is really to report that to the department and then they do the follow-up for that. So, I see someone's popped in the chat. Thank you, Jason. And I can have a look at that article. I wasn't aware of that this morning. But yeah, so basically if you see something that is suspicious, report it to the state or federal department. And often for things that are highly damaging potentially or are increasing the risks, then there will be usually a case already in progress and they'll be collecting evidence. So that's what it looks like. This looks like it's happening for seeds already. So, but if there's any comms that we can mirror, I'm sure that we can talk to the comms office with Brad and Sam, and we can probably put that into some of our e-newsletters as a warning as well.
Yep, thanks, Nicole.
And Megan McKenzie's just popped up a link as well for the National Exotic Plant Pest Hotline.
Thanks, Megan. I was going to mention that, but I couldn't remember the number off the top of my head.
Okay, and don't forget that you can contact Kevin, Nicole or Rob with any further follow-up questions. Their contact details are on the website or you can get their details from myself. So feel free to reach out if you've got more questions, and we might wrap up the webinar here then. So thanks to everyone for coming along, and thank you to our presenters. It's been an excellent series on biosecurity.